Good morning, everyone, and thank you for joining us for today's webinar. Today's webinar is entitled DNA Methylation-Based Age Prediction and Body Fluid Identification. We have two wonderful presenters today. Our first presenter today will be Dr. Athena Vidaki. Dr. Vidaki currently is working as the postdoctoral researcher at the Department of Genetic Identification at the Erasmus University Medical Center in Rotterdam, in the Netherlands. She holds a Bachelor's of Science degree in biology from the University of Athens, which she received in 2009, her Master's in Science in Forensic Science from King's College London in 2010, and she also successfully completed her PhD there in forensic epigenetics in 2015. Her study on age of donor prediction as part of her PhD was awarded the prize for the best presentation of the 2015 Congress of the International Society of Forensic Genetics, and she also recently won the Young Investigator Award in 2017 in the field of forensic genetics, which was awarded by the International Society of Applied Biological Sciences. Our second presenter today is Dr. Huanyang Li. She received her PhD from the Yonsei University in 2001 in the field of biochemistry. Following 1 year of postdoctoral study at Protein Network Research Center in Yonsei University, she moved to the Forensic Medicine Department at the Yonsei University College of Medicine in 2003. The most recent interest of Lee is to expand the application of DNA methylation profiling to the analysis of evidence samples for investigative leads such as age prediction, where lately her group has reported high accurate age predictive models for forensic relevant body fluids, saliva, and semen. Our disclaimer: the Forensic Technology Center of Excellence, the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now, before I hand things over to our first presenter, I would like to hand things over to our moderator, Dr. Daniele Podini, for some words introducing today's webinar.
Thank you, Josh, and thank you to all our attendees. I should say good morning, good afternoon, and good evening to them, because there's people from all over the world. We can truly say that the sun never sets on this webinar series. I feel this is very exciting. Today's webinar is not technically about SNPs, despite the series is all things SNPs. In fact, what is used to predict age and identify body fluids in this case is DNA methylation, which is not a change in the nucleotide sequence. The reason why I felt I could cheat and include this topic in our SNP webinar series is that methylation, as you will hear soon from in greater detail from Dr. Vidaki, is that the methylation is detected by a chemically induced sequence change where a non-methylated cytosine is converted into uracil and then through PCR into thymine. So in essence, it is detecting a SNP. So that is why I felt legitimized to include this topic in the series. So now you've heard my justification. Without further ado, I'll let Dr. Athena Vidaki begin her presentation. And thank you again to all.
Hello, everybody, also from me. Before I start, I would like to thank Josh for this wonderful introduction and also Professor Podini for this invite. I'm very excited to be here today and that I've been given this opportunity to talk to you about my work on DNA methylation-based age prediction and how this can be used in forensic applications. So I hope you can all hear me well, and I will continue with the slides. So during the standard forensic DNA profiling, we know that every crime starts with a crime scene, where biological evidence, such as this blood stain that you see on the screen, is collected for being processed for the normal DNA profiling using the short tandem repeats. Now, once we are successful and obtain a nice STR profile, The standard procedure includes comparison between the DNA profile from the crime scene and the DNA profile from the suspects we have, or run it through the national DNA databases. If we get a match, then the prosecution is possible based on this DNA evidence. However, when we don't get a match, most of the forensic investigations can lead to a dead end. We have an STR profile, but we cannot locate the donor of this evidence. So in these cases, forensic DNA phenotyping, which has been developed over the last few years, including a lot of work in the department I'm currently in, can help in those cases. So the police often asks questions, for example, where the stained donor come from, and also clues on their appearance, including the hair color, how tall the person is, skin color, eye color, and so on. And the how old is the person is actually one of the questions that often comes up and can help the police target their suspect pool by minimizing potential suspects. And this is what I will be talking to you today. So as I just told you before, for eye, hair, and skin color, there are existing tests that you've also heard as part of this webinar series a few weeks back. That are based in a small set of single nucleotide polymorphisms that can actually generate results that provide high prediction accuracies. Those characteristics can be also associated with the biogeographical ancestry, where we also have tests for the prediction of that. And future work will show an improved accuracy by including more and more markers. However, when we go to more complex questions, such as how tall the person is, or what do they look like in terms of facial morphology, researchers soon realized that it will be very difficult to do so with only a small set of markers. In those scenarios, probably we are talking about hundreds or even thousands of markers. Now, as you will see in this small animation here with a small child becoming an old woman, you see that predicting the age of the person can actually also assist the prediction of other characteristics, such as, for example, the skin morphology or baldness or facial morphology that are actually also age associated. So in this presentation, I will give you a little bit of an overview first on the current and existing biological methods for age prediction. As we know, also in the anthropological field, we have age prediction methods using the bone structure or the bone morphology, but I won't be talking about this today for time reasons. However, I will give you a little overview of what had been done before epigenetics came into the field. Telomere length, mitochondrial DNA deletions, and T-cell DNA rearrangements have all three been proposed for age predictions. For example, telomere length is believed to decrease by 20 to 60 base pairs every year of our life. However, these techniques are not very accurate, have been only tested in a limited number of tissues. And as an example here, I have the prediction model by the paper of Supakov in 2010, where he used the T-cell DNA rearrangements to predict age, but the standard error was actually quite high with almost 9 years. Meaning that a person could be predicting either 9 years older or 9 years younger, meaning that we are still having a 20-year gap. For a preliminary study, this is great, but it was soon evident that a marker that can be very nicely correlated with biological age, hence with also chronological age, is also DNA methylation. But what is DNA methylation? DNA methylation, as you can see here, is one of the mechanisms of epigenetic mechanisms. Let me use the arrow here that are found in our cells. Together with DNA modifications, including not only methylation, but also hydroxymethylation, we also have protein interactions, including nucleosomal remodeling, meaning how the chromatin is remodeled on the DNA, non-coding RNAs, which is also explored in forensics in other applications, and also modifications on the histones, the proteins that can be found to be wrapped around the DNA, including a wide range of this. For the purpose of this webinar and for this application, for now we have only focused our efforts into the DNA methylation. But where can DNA methylation be found? The answer to this is in the 5 position of the cytosines that are only followed by guanines. So you can see here that in the 5 position of the cytosine, we have the possibility of having a methyl group, but only for the cytosines that are following by guanines. Now, this is the rule, but we know in research and in biology that these rules can be changed over time. So for now, we know that the majority of methylation is found in those so-called CpG sites, but we will see in the near future that this might expand also in other bases. When we take the genome as a whole, we will see that most of the CpG sites are methylated, 60 to 90% depending on the organism and the cell type. However, and very interestingly, most of the unmethylated sites are grouped in so-called CpG islands, which are locations which are very high in GC content, and there happen to be at the promoter region of many genes. So this observation already pointed towards a kind of role in gene expression regulation. How does methyl group being added here? Well, it can be added via either maintenance or de novo methylation, meaning either kind of inheriting methylation patterns after every DNA replication cycle, or de novo methylation to set up new patterns, as also respond to environmental factors. So in these procedures, we have different enzymes involved. As I mentioned before, we know the role of gene expression here. By default, again, as a big rule, when we have a methylated gene, usually the gene is not expressed, so it's inactive. However, there are also exceptions to this rule. As you can see here, if we are talking about CpG islands at the 5' end of the gene, the rule says that when the promoter is unmethylated, the gene can be expressed and can lead to an mRNA and a protein, whereas when it's methylated, it blocks the gene expression. And this usually happens by the recruitment of binding proteins, where they come to the DNA, they wrap it around it, and they make it non-accessible to the transcription mechanism. If we have a look closely to the DNA strand and how the methylation is formed and changed over time, you can see here one example where, as we said before, methylation can be found only on these CG sites. When we are born, we inherit those methylation patterns from our parent cells. And an example of a profile can be found, a very simplified example can be found here, with some of the CpG sites being methylated, whereas some of them can be unmethylated. Again, this is just one strand, so one cell. However, as we grow and as we are exposed, the cells are exposed to different environments and different factors, including our diet, our metabolism, our medication, if we're smoking, if we're drinking alcohol, if we're exercising. All these factors can affect DNA methylation by making those methyl groups either the CpG sites either lose or gain methylation. And as you can see here, there are some sites where they stay methylated or unmethylated throughout our lifetime because they are involved in very important processes. But there is also the set of CpG sites like those two, where they can lose or gain methylation. And this differential methylation actually is what we care about in forensic applications, but also in the medical field. Now, when we go and we talk about epigenetic-based age prediction, there are a couple of epigenetic clocks where they're very famous and people can have been using them mainly in medical research that I would like to present you here. before I go into the forensic models and forensic studies. This one was the paper by Hanum, published in 2013, where they used 660 samples aging from 19 to 101 years. They used the Illumina 450K platform, which is a microarray allowing for the analysis of 450,000 of those CpG sites in our genome, so almost 500,000 markers. And they based their analysis in whole blood. So what they wanted these people to do is to select from those 500,000 sites a subselection of CpG sites that can be used for predicting the aging rate. And as a result, you can see here, if we plot, let me also use the arrow, if we plot the individuals from 19 to 101, you can see all these 500,000 sites. And by looking at blue when it's kind of unmethylated with a high fraction of methylation, you can see that these methylation patterns actually do change for some of these sites, and they show kind of a gradual methylation loss or a gradual methylation gain. Using 71 of those sites, they managed to build a very accurate model with a standard error of 3.88 years. And what they also investigated here is the fact that there are some individuals that are healthy by default, in a healthy state, which they either age slowly or age faster. They also noticed that there are some differences between females and males, as you can see in the graph here. And around they proved that males, unfortunately, age 4% faster than females. So this was a very interesting study, blood specific, and it was followed up also a year later by another study trying to minimize the 71 markers down to just 3 CpG sites. Originally, they had a similar setup with the previous study, but in this case, they used the previous platform, the 27K, which allows for 27,000 CpG sites only. And they they identified 102 of those age-related CpG sites in their dataset. Using these 102, they also built a model here with an improved 3.34-year error. However, as you can also see, there is a high representation of older people, which is not ideal in such models, due to the type of studies that they used to pull their data, which were actually investigating age-related diseases. However, from 102, they picked the best 3, and they run 150 more samples to creating this epigenetic aging signature using prior sequencing. And actually, the model that they built was not actually that bad. Of course, as expected, by reducing the number of CpG sites, the error increased by almost 2 years, but they actually got a less error in their testing set. So this study showed us that yes, it's possible to use just a few numbers of CpG sites to predict age, at least in the decade level, meaning that we can say if somebody is in their 30s or in their 40s. However, this is restricted to blood only, so it's tissue restricted. Koch and Wagner published also in 2011 a study where they tried to go beyond this restriction on tissue type or cell type by investigating 13 different cell types, again in the 27K platform, and with almost 900 samples of various ages. From their study, it was evidence that there were 5 CpG sites that they were actually the ones that showed the most significant correlation with an age prediction of 11 years. Here on the right, you can see the methylation graph. So this is the methylation levels from 0 to 100 and the age from 0 to 80. And you can see how methylation changes over time. They have labeled different tissue types with different colors. So you can see that different tissues, such as this one here, for example, breast tissue, has different methylation levels and reacts differently over time, which I think is also a big limitation of this study because we can see that there are some tissue and batch and study effects that can be part of why the error was actually so high. And here we have the model based on 5 and based on 3 CpG sites. And you can see that the correlation is good, but it can also be improved. And the one I will talk to you last is probably one of the most popular studies done by a single author, Horvath, in 2013. And you will understand why. Professor Horvath from the US collected publicly available data from a lot of different studies. I think there were around 80 studies. He pulled them together and created a very massive data set of almost 8,000 samples. Working on this, in order to be able to get an 8,000 samples, he decided to include studies that have used both the 27K and the 450K platforms.
All guests have been muted.
And as a result, he could only identify and investigate 21,000 CpG sites. Once he did that, he ended up with 353 so-called clock CpG sites that he investigated in a total of 53 different tissues. The model using all 8,000 data, this is the results that I'm presenting you from the validation test, gave very high prediction of 3.6 years with a great age correlation with age of 0.96. However, if we look more closely to individual tissues, we can see that of course, as expected, the predictions slightly change. Here I have chosen 4 of them that could be also forensically relevant, including whole blood, buccal cells, saliva, and uterine cervix for vaginal cells that we often have in forensics. And you can see here that the correlation and the prediction is quite different. As you will also see, especially in the example of buccal, the age range that he could include in this analysis was very small. So we are talking about very, very young children. So the study that was that the data was available for this purpose included only very young individuals. So that's why perhaps the error looks so impressive of 0.37, but it is a matter of further investigation to see how these sites develop over time for buccal cells. Saliva also here, and you can see also the limitation of having data and samples of the same age and not kind of a continuous model here. But in any way, this study was great because it proved that we could have multi-tissue predictors based on what it's considered in the medical field a small set of samples. It's only 353 out of the 500,000. But of course, we know that in forensics, this is very high number, and we often are not able to get this into from forensic stains. Also, this study was only based on the microarrays and was not followed by targeted technique, which we aim to do as part of this project. So a little bit of a background on epigenetics in forensics. This is a very new subfield, especially DNA methylation, however, a very fast growing. So I started this work in methylation back in 2011 and published this nice review in 2013, kind of showing the potential opportunities there, but also some initial challenges that the forensic applications will face. And since then, we have much more, which you can go and have a look if you want to kind of expand your knowledge in this field. Mainly, we have so far successful studies in 3 different applications, including which tissue or which body fluid. I have also published in this, but Professor Huang Yang will explain everything about it in the follow-up presentation. How old the person is, which I'm talking to you today, and also DNA methylation can also be successfully applied for which twin is the stain of the donor, which we have started also investigated here at the Department of Genetic Identification. So these three applications. When we want to take, no matter what application we are talking about, when we want to apply DNA methylation profiling in forensics, we have to have in mind a set of forensic challenges which are very important for successful implementation. First of all, in forensics, we are always looking for accurate prediction, at least as accurate as possible. We are dealing with forensic samples of both low quality and low quantity, meaning that we need to use as small number of CpG sites as possible, and it also means that we have to have as sensitive methods and reproducible as possible. Those samples usually have unknown parameters related to them, meaning that often we do not know the tissue of origin, or we don't know the time of deposition, or we don't know the environmental effects that these stains have been exposed to. We also have limited information on the health and disease state of the individual, and as you saw in the animation before, all this variable DNA methylation can be affected by different health and disease states. Therefore, we have to investigate all this with a goal to create a statistical framework later on that we can take the results that we obtain and we can make it to a meaningful prediction for the police. Going into the forensic studies, the age prediction research already started in 2015, maybe 2014. I have a selection of studies here, with the LOVL2 gene being kind of the king of these age-associated genes. So a group in Poland investigated 300 samples of various ages using prior sequencing, which is a technique that had been used before for other forensic applications, mainly in SNP analysis. But as Professor Podini mentioned before, the methylation turns out to be a SNP detection. Therefore, people thought that it can be also used successfully for methylation analysis. They investigated 7 CpG sites in one genomic region of this gene, and their results were quite impressive, with a prediction error of just less than 7 years using only 2 CpG sites. This is the correlation between chronological age and predicted age as they saw it in their data set, and you can see they get a very nice correlation of R-squared value of 0.86. They had also tried initially to test a little bit of some common parameters in forensics, such as how many years the stain have been deposited, and they saw that if the purple is the successful PCR reactions of the total samples and blue is the correct predictions, you see that over time, actually the success of the correct predictions is lowered because we indeed have the environmental effects, and it's actually quite expected. However, digging more precisely into age groups, they also saw that while they were predicting very highly in young or middle ages, when they were talking about investigating people 60 to 80 years old, the correct predictions were much lower. And this also goes to the fact that those CpG sites detect more the biological age, meaning how fast our cells age, rather than our true chronological age, which is our date of birth. However, they followed up those very impressive preliminary results with investigating a total of 8 loci and 41 CpG sites in a much larger sample. The same technology, which is great, but I would like to have you in mind that it's only currently used in the singleplex mode, meaning that you would have to investigate each of these genomic locations separately. However, they managed to pull the 5 CpG sites that belonging to those genes that were the most significantly associated with age and end up with a prediction error of 4.5 years. Again, you can see that they still had the problem of going for the larger category of 60 to 70 years old. They had actually only predicting only half the people correctly. Since then, there have been many studies, which I could not mention all of them, but I would like to finish before I start with our results with this epityper system, which is a new system to be used for such analysis for age prediction. So this was a very nice study by Freira Araves and colleagues published last year, taking this a little bit further, focusing on Europeans only, in 725 Europeans, and looking at, again, only 7 DNA methylation markers that gradually lose or gain methylation to be able to predict age with less than actually 4 years. And together with this, they had used the SNPer forensic classification system to create an online tool where you would run your samples and you would put the methylation values and you can get the predictions afterwards. This is actually a nice study showing also the application by other laboratories and how this can be done. When I started this work in 2014, we had kind of 2 aims in mind. One would be how low can we go in terms of age-associated CpG sites, and how sensitive and accurate we can go using the very fast-developing methodology in forensics now, massively parallel sequencing or next-generation sequencing. So we had acquired the MySeq instrument in the lab, and I was very keen to try to see if we can use it for such methylation protocol. And later, since we got very nice results, we were also wanted to start a little bit on the validation side and see how we can kind of assess both accurate prediction, but also analyze forensic type material. We picked this study, as I told you, because it was the most robust and the most, it included the most samples, and it had the possibility of having CpG sites that were multi-tissue applicable, with a 3.7-year error in whole blood, and focus our analysis in a subset of those sites. So what we did is we selected 45 of them, showing either the strongest positive or the strongest negative correlation with age, meaning either gaining or losing methylation over time. And we wanted to focus our analysis in blood, but with the potential of applying this to other tissues. Therefore, we created our own data set with some of newly publicly available data that we had at the time. And we created a sample set of 1,156 samples ranging from 2 to 90 years old. We tried to have a very good representation between our age groups to avoid problems that I've showed you before, with around 100 samples per decade. And what we did is we checked how methylation changes in our data set. So here I have an example of 16 CpG sites with their methylation levels and how they change over time. And as you can see here, we get either CpG sites that lose methylation or gain methylation, or some CpG sites that show very minimal effects. Doing some statistical testing, we managed to get significant p-values for 25 of them. So we refocused our analysis into these 25. We also used multiple regression and stepwise analysis to select the best of those 25. And we proved that with stepwise multiple linear regression, we saw that after the 23rd CpG site, the predictions actually were not changing significantly. Therefore, we managed to create a tool with 23 CpG sites based on multiple linear regression and with a prediction error of 4.61 years. This gene, the CpG belonged to this gene, showed to be the most significant one, but we were very keen to try to kind of test different modeling approaches and strategies to see if we can get this error a bit lower. But in general, we had 61% of our individuals being predicted within 5 years. So in the department at the time, we had researchers working with computational methods, which the last couple of years seem to be a very hot trend in medical research, the so-called artificial intelligence or artificial neural networks. So these are computational methods that resemble our neural cells and can recognize very complicated nonlinear relationships with certain variables we choose. So we thought that especially those CpG sites that do not seem to change in our eyes very much, perhaps they can contribute in age prediction with a nonlinear way of relationship. And what we did actually is we selected a subset of 16 CpG sites of those 23. We used generalized regression neural networks, and we were successful in creating a model that was accurate in our training set of 3.3 year of prediction error. So we both lowered the number of the CpG sites and also used a different analysis strategy, and we almost did this 1.5 years less error. We used a quite conservative approach, meaning that we use 60% of our samples for our training of the model, 20% as verification set, and 20% as complete blind test, corresponding to 231 samples. As you can see here, this is the model. The correlation was pretty high with 0.9637. And our error, even in the verification and test set, were actually below 4.5 years. However, when we look at the residuals, which is in this graph here, we see that when we have older individuals, the prediction error actually tends to spread. And we see some bias towards underestimating methylation. So this is something that can be worked on a future model, how to improve this and therefore improve the mean average prediction error. Also, if we look here at how much each marker contributes to the model, this is the error that we get when we take out individually each of the CpG cells out of the model. We see that the error significantly increased when we take out this marker here, or this marker, or this marker, but there are some other markers that do not contribute to the prediction that much. So an idea would be, can we take these less important, if I can say, markers and still get quite acceptable predictions below 5 years? We spent some time for some initial model validation. We were very interested to see how the health and disease status can affect prediction. So we created a data set of almost 1,000 samples, just over 1,000 of various different diseases, and we could see that the error actually gets worse especially when we talk about diseases that have to do with blood or bone marrow, by having an average error of 11 years. Of course, this is something, as I said, that you do not know of your stain donor, so we should create kind of a strategy how to deal with these possibilities. We also tested 265 saliva samples, and we were very happy to see that even with a small data set, we could replicate similar prediction errors of 4 years in our test set. So this could actually lead to our observation that yes, this model can be used perhaps in other tissues beyond blood. If we look at those 16 markers closely, we will see that some of them are being involved in diseases such as cancer or type 2 diabetes or brain damage, some age-related diseases which one would expect, but can affect the biological aging, hence why we saw the lower prediction errors in our disease data set. And that was the part one. So we were pretty happy to say that yes, a small number can be used for this purpose. Going into part two, we were interested to develop a method that can be as easy as possible, as accurate as possible, and as reproducible as possible. Every method, of course, starts with their state. So we focus our analysis on blood samples. From that, we would extract the DNA. We would then perform the DNA quantification using quantifiler as a standard forensic sample. And then we would perform a step called bisulfite conversion. This is what Daniele talked at the beginning. This is a chemical modification reaction. This is the most popular way that researchers have to translate differences in DNA methylation into differences in sequences, because we want to have a PCR-based test, meaning that we need to kind of have to deal with sequence variation rather than with methylation variation. So during this process, we use a chemical called sodium bisulfite, where it has this unique ability to leave methylated cytosines, CpG sites, untouched. Kind of the methyl group works as a protector, whereas the unmethylated sites will be transformed into uracil. And hence later, when we are amplifying the genomic regions of interest, we will convert those uracils into thymines. So we would end up with a CT variation similar to what you would do with a SNP. However, I would like to bring your attention here that it's not as easy as it sounds, because bisulfite converted DNA templates are very difficult to work with and amplify. This is because of the reduced complexity of those DNA strands, because cytosines that are following by other bases such as A or T will also be converted. Therefore, our template, we're going to be very T-rich. Therefore, the design of the primers can be very challenging. So once we perform the bisulfite PCR, we would pull everything together. We would quantify to see how much we have. And then we would begin the procedure of the library preparation. As I said, we use the MySeq platform. So we tried this with a TrueSeq protocol, quantify our libraries and run it through it and see how successful we can be with the methylation detection. We used some artificially made, commercially available DNA standards for this, and we were pretty happy to see that the methylation quantification was pretty linear. However, we used this as a standard curve to normalize our obtained data to be used for age prediction. Some initial validation, which is currently also being continued by my colleagues at King's College London. We have kind of, as I said, a pretty nice linearity curve. However, as you will see, those standard deviations here, those bars, look pretty high. The reason for this is mainly because this is the pooled graph, meaning we take all samples and all markers into account. If we look specifically at each marker, we will see that we get markers that we detect very nicely and very linear in a linear way. However, we also get markers that show some preference towards the methylated allele, so we kind of overestimate methylation, or some markers that we underestimate methylation, meaning, for example, that our 50% standard, which should be here, we should detect it somewhere here, but we detect it to be something around 30%. So during PCR, we have some, or during even library preparation, we have some preferential amplification towards the unmethylated allele. Initial experiments also showed that this can be very sensitive with only 1 nanogram input into our library prep, or reproducible by running a same sample various times, 6 times in this case. We could predict somebody as being 56, where his true age was 54. However, when we increase the samples into our MySeq and run it through the model, we realized that the mean error we are getting was much worse than we were hoping for, meaning that we had to go deep into more technical variability. The main problem that we believe is the reason for this 7.45 years based on a limited number of 46 samples is because we use NGS-based data and we use a 450K based model. This is a targeted technique, this is a microarray technique. There are differences in the technical variability there, and they can introduce different detected methylation levels, meaning that these predictions are quite skewed. So we were very keen into try to optimize this method to have as less manipulation steps as possible, leading hopefully to more accurate predictions. So what we did is we further reduced the number of CpGs from 16 to 12. As you saw, there were some markers that seemed not to be contributing to the prediction that much, so we took them out. And we created, together with an MSc student at King's, 2 multiplex reactions, a 7-plex and a 5-plex, to kind of also increase the forensic applicability of this by running only 2 PCRs rather than 12. We also adjusted our pooling strategy. because we saw that in the MySeq reads, some markers were represented higher than others. And of course, it is a number game here. The more reads you have, the more accurate the quantification will be. Therefore, we tried to improve that by adjusting the way we pulled the markers together. We also did adjust the library amplification, because we thought that by decreasing the number here, we would decrease also the extra amplification bias. And we were very happy to see that actually those results did make a difference. We also wanted to adjust for the differences in the technology, and we wanted to see if we can run a lot of samples here with this method and kind of get to a high enough number to rebuild the model based on NGS data only. And just before I left King's to move to Rotterdam, we had some very positive results of how to predict, how to build an NGS model based on those 12 sites only using the MySeq platform. And we were back actually to our usual, if I can say, errors of below 5 years. Of course, this is ongoing work. We need many more samples to increase this to be used. But I would like now to move to part 3, which is a little bit more into the validation phase through an exercise via the European DNA Profiling Group. When we presented this at the ISFG in 2015, people were very interested to test this themselves in their labs. Therefore, we were very happy to organize this exercise with 14 participating laboratories from all over the world. And we wanted to kind of expand the participants by including not only the MySeq platform, but also the IonTOR PGM. We split the design into 2 parts. First of all, we sent the laboratory 7 standards just to familiarize themselves with the technique. Most of this laboratory did not have DNA methylation experience before. Therefore, we wanted to get them going. And then the part 2 would be to send them 7 blood stains with optional analysis of additional samples, and then see how the prediction looks like. The results of the part 1 were quite positive, but also led us to some interesting observations. We saw that the MySeq laboratories, if you see here, are the MySeq laboratories here with the PGM here. I'm sorry, there seems to be some issue with the sound from my side, so I hope you can hear me well. I will try to speak up. You can see that with the MySeq platform, the labs were getting reads from 100,000 per sample up to as high as 700,000. However, we had differences between laboratories as expected. When we looked at the PGM, we had similar variation. However, we saw that the number of the total reads we were getting per sample were actually almost 8 times less. One can expect that, thinking that the protocol had been optimized with the MySeq platform, whereas it was the first time trying the PGM with different chemistries and different sequencing technologies. Looking at reads per marker, we had a similar observation here. Even if we had optimized the pooling strategy, we still saw that some markers were performing very poorly compared to other markers, and there was also a difference between MySeq and PGM. Depending on the marker, we could have, with the MySeq, 3 to 41 times much more coverage compared to PGM. Looking also individually per lab, only focusing now on the MySeq laboratories, we see some variation here. Remember, everybody started with the same sample, with the same protocol, but we see that we could have differences explained by the different operators or the different systems and the different kits that people used. However, for most of the CPG sites, no matter, regardless that PGM resulted in smaller coverage and perhaps 10 times less reads, we saw that the linearity graphs were very good between the concordance between the MySeq and the PGM. There were, though, a few set of markers where there were differences as a result of the less coverage, which could affect our methylation detection and also the age prediction. So the outcomes of this part 1 was that people could perform this even if they had no experience with DNA methylation-based NGS, which is very positive. PGM performs good, but we should optimize that further if people want to use this platform. Overall, though, we got a standard deviation of 3.4 in our methylation detection, which is very optimistic for future implementation. We also saw that the hardest samples were the ones showing lower methylation levels, which could indicate that these samples are more prone to amplification bias. Together with Myra and Professor Parson from the Medical University of Innsbruck, we tried to spend some time optimizing this PGM protocol before we move to part 2 of the exercise. This is ongoing work. We are currently writing this up and hopefully publishing this soon, but I can give you a small overview of the prediction results of the part 2 of the exercise. With purple, you can see the PGM labs. With blue, you can see the MySeq labs. And you can see that most of the MySeq labs, at least, get 4 to 5 prediction error figures similar to ours. However, as a result of a less optimal PGM performance, the prediction was 2 to 3 years higher. We could see also some individual differences per lab. We could see labs that could predict everybody within 10 years of error, but we could have instances of labs of kind of a more random prediction here, which could indicate extra technical variability during the procedure. I mean, what I want people to have in mind is that this is a quantification method, and any manipulation and any normalization can really improve those results. So lastly, if we go back to the question of how old someone is, and you ask me if this is possible, yes, it is possible, but we still have some way to go to kind of improve the technology before implementation into casework. And what we could do is on various different things. Part of some ongoing work we do here, since we realized that actually we only need 10 or 12 markers or even less to do age prediction, is can we translate this into a simpler, cheaper, and more accessible method using a method that you will hear a lot by Professor Huang Yang later, a methylation snapshot, and some initial results are very optimistic. By translating a subselection of those CPG sites into kind of a CE-based method rather than an NGS. However, of course, NGS has, you will agree with me, great advantages because it can be used be combined by other tests in a more kind of massively parallel sequencing approach. So we are still working also on this. People and other research groups have been doing a lot of nice progress of testing different tissues, such as the group in Korea on semen or on saliva. People have also tried to improve age prediction by combining skeletal and dental ages and focusing on specific age groups such as children. We have also the group trying to apply this into more complex and challenging samples such as human teeth, or also a recently published paper trying random forest regression, so other types of computational methods and other types of prediction modeling, which can further improve our outcomes. And I truly believe that we need kind of combine the forces and see the results of everybody and choose eventually the best approach for such application. I would like to summarize here now by kind of put a little bit of perspective of what I've presented you today. Age prediction based on a small set of markers is possible at the moment at the kind of plus/minus 5-year error, but I envision that later this error can be reduced by including more sites, or by targeting our research into more age group specific markers. We saw differences in people in older ages, so perhaps we have to adopt our modeling approach to target those groups. Most of the current forensic literature is indeed tissue specific. We have models that work really well in blood. We have tissues that work, models that work really well in saliva. And I wonder if in the future we could also combine this and find markers that can lead us to a universal tool. As you saw, people have started trying different tissues such as skeletal remains. We can open up the applications and how useful this age prediction test can be. So more challenging tissues could be also a future investigation topic. And lastly, MGS is useful. We know that methylation quantification can work. However, we did see differences between the platforms. Therefore, we would need to focus our analysis in the best platform or the platform that we have in our lab and really validate it, forensic type validation, to really understand how each method performs, including, of course, sensitivity and reproducibility testing. And lastly, we still have to investigate various parameters such as population differences. One might think that age markers in European populations might not be the same with an Asian population, but also assess how our markers are being influenced by a disease state. Lastly, I would also like to announce the VISAGE project, which corresponds to the Visible Attributes Through Genomics Consortium, which was recently funded by the European Union's Horizon 2020 program. Professor Manfred Kaiser here at Erasmus University Medical Center is the coordinator of this project, and we have 13 different laboratories involved, including 6 police forces. What this 4-year project aims is to take established knowledge and develop genotyping and statistical tools that can be forensically validated and straight implemented into forensic practice that will not only predict age that I spoke to you today, but also appearance and ancestry from small DNA, forensic type DNA traces. They will also study ethical, societal, and regulation dimensions to these predictions and kind of put this forensic DNA phenotyping field one step forward. I would like to finish the presentation with this slide, kind of as a take-home message. I think epigenetics has a lot to offer in forensics. I've talked to you about before about the 3 main applications that we have been using it so far: tissue ID, age prediction, and differentiating between twins. But we really believe that the applications are still many and they will come in the future. And we have just recently, actually this week, published, it's currently in press but should come out soon, an opinion article in Genome Biology where we discuss some of the future potentials of forensic DNA methylation profiling to kind of answer many different forensic questions, including lifestyle prediction. Can we say if a donor is a smoker, if they drink, if they use illicit drugs, and so on, as you see on the screen. So if you're interested in finding out more about this, please follow up with reading the paper. I would like, of course, to thank everybody involved in this project. I could not have done it without them. My colleagues at King's College London, especially David Ballard and Denise Cinder-Concourt. Most of this work was part of my PhD project. Of course, Professor Manfred Kaiser and Hedayat, the student that is currently working with me on the age prediction. Professor Parson and Myra from Innsbruck for their help optimizing the PGM protocol. The European DNA Profiling Group and all participating labs for their great interest and collaboration in this EDNAP exercise. The University of Athens and Papadaki Foundation for funding my PhD project by giving me a doctorate fellowship, and also the Euroforgen Group for supporting an exchange visit via one fellowship. And last but not least, my group at Erasmus MC, which makes my everyday life really happy. And I would like to wish you all happy holidays. And if you have any questions, I will be very happy to answer them either at the end of the webinar or via email. And I would like to thank you again and move on to Professor Huan Yang.
All right, thank you so much, Dr. Vodacki, for your presentation. We had quite a few questions come in that we will answer at the very end of the webinar. Dr. Huan Yang will be sharing her screen all the way from South Korea today. While we bring that up, You should see her presentation populate here just momentarily. And Dr. Huang Young, I'd like to go ahead and hand things over to you and let you get started with your presentation today.
Hello everyone, thanks for attending this talk. The topic of my presentation is DNA methylation-based body fluid identification. I hope this talk will be helpful in understanding how multiplex assay targeting CPGs can be used to determine the body fluid of origin from a DNA sample. We will see the following 4 parts. Firstly, body fluid typing in forensics to see how it is initiated and its characteristics. And secondly, identification of body fluid-specific CPGs that demonstrates how it's chosen as predictive biomarker for each body fluid. Thirdly, a snapshot multiplex as a technique or kit that we've been using. And lastly, several examples of case drug analysis, how it is utilized and aided in police investigation with higher possibility. Let me start off by introducing implications that body fluid identification has in the forensic field. Body fluid identification or body fluid typing refers to the determination of the type and origin of body fluid. Body fluid identification has a significant implication because of its possibility that links sample donors with actual criminal acts. That is, the presence of certain type of body fluids can be used as an excellent indicator of the sequence of the events which have occurred at the crime scene. Blood stains can indicate some form of physical struggle, assault, or murder, and detection of semen or vagina secretion can indicate the involvement of some form of sexual encounter or assault. Menstrual blood and saliva may be proposed as an inoffensive alternative scenario to an alleged violent assault. When internal organ tissues such as brain were found on the clothing of a suspect and then match the DNA profile of a victim, it can suggest that the suspect was in the vicinity of the victim during the crime. So, when a potential body fluid is discovered at the crime scene, a particular form of light or chemical addition may be required to visualize the stain that is difficult to see with the naked eyes. As you can see here, techniques which are currently used for forensic body fluid identification include chemical tests, immunological tests, protein catalytic activity tests, spectroscopic methods, and microscopy. Some of these methods are presumptive tests which are used as a screening test, such as the luminal for blood and a CP test for semen. Others are confirmatory tests that will conclusively identify the presence of a certain body fluid, such as the microscopic identification of sperm cells. However, when it comes to the detection of vaginal secretion and menstrual blood, there has been no available method until recently. Moreover, there are several limitations in most conventional body fluid typing methods. These limitations include low specificity, lack of sensitivity, sample destruction during the test, instability of biomolecular assay, or incompatibility with downstream assay analysis. In addition, all of these methods are carried out for only one body fluid at a time and cannot be applied when only DNA extract remains in an old case. Such being the case, molecular approaches that can overcome these limitations have been intensely studied. Then how can we detect and discriminate different types of body fluid using molecular methods? Different tissues and body fluids contain different types of specialized cells, and cells become specialized by selectively activating different genes within the genome of the chromosome. There is determination of the cell type may occur at the level of gene expression, which commences with the transcription of DNA into RNA and proceeds to translation to proteins with specific functions. Epigenetic modification that can alter the local chromatin structure and the presence of microRNA may also affect cellular specialization through gene expression regulation. Therefore, specialized cell types carry distinct RNA signatures and different epigenetic profiles as well as different functional proteins. However, let me put more focus on epigenetic variations rather than RNA signatures because we are interested in the development of DNA-based approach. Epigenetic profile is a collection of chemical tags such as DNA methylation and histone modification. And as you can see here, DNA methylation profiles are specific to tissue and age. Tissue-specific DNA methylation has also been investigated in human autopsy specimens. If we take a closer look at the cluster analysis in this slide, we can see that the tissue from different individuals show the highest concordance of DNA methylation patterns compared with other factors such as person, diagnosis, gender, age, and race. Brain or spleen tissues from all the 6 autopsy cases clustered together, and the heart, kidney, or lung tissues from 4 or 5 autopsy cases clustered similarly together, which implies DNA methylation patterns are more consistent between the same tissues from different people than between different tissues from the same individual. This slide shows the most common features in which DNA methylation varies. DNA methylation at a single CpG site is known as a methylation variable position, and the region defined by increased variability rather than gain or loss of DNA methylation is referred as variably methylated region. More noteworthy is that the region called differentially methylated region exists within the genome, and at this region, multiple adjacent CpG sites show differential methylation depending on many different contexts such as imprinting, tissue reprogramming, cancer, and age. In the forensic field, the potential of tissue-specific differentially methylated region has been examined for the identification of body fluids. This paper first demonstrates the application of the testis-specific DMRs to the identification of semen. And to date, many papers reported body fluid-specific DNA methylation and its application to forensics. Some reported multiplex assays for the detection of body fluid-specific CpG markers, and others validated these markers and multiplex assays. This is a snapshot multiplex that I'd like to introduce to you today. This multiplex is composed of 9 CpG markers, SA1, SA2, BL1, BL3, BF1, BF2, SA1, MB1, and MB2. They show a methylation signal only in a target body fluid and enables to differentiate among semen, blood, vaginal fluid, saliva, and menstrual blood. In this electrophoregram, methylation signal is shown as in blue and non-methylation signal is shown as in green. So a visual check of the presence of the blue methylation signal allows us to determine which type of body fluid is the source of DNA. For example, if a DNA sample shows blue peaks only at 2 blood-specific markers, BL1 and BL3, we can say that the DNA was originated from blood. Then I will move on to talk about how to develop a multiplex assay for body fluid identification. Assay design means the development of local specific analysis for the detection of single or a few genes that are already established as predictive biomarkers. Therefore, selecting relevant marker is essential. In general, relevant markers can be selected from studies employing epigenome-wide technologies. Among many methods, bisulfite microarray is the most public and easily accessible, and now enormous data generated using Illumina's village array can be browsed and downloaded from the public database. However, since no body fluid data except for blood and saliva were available from the public database, we had to produce DNA methylation profiles using bisulfite array and select body fluid specific marker candidates. Then we further tested the body fluid specificity of the candidate markers using targeted bisulfite sequencing. Once the markers to be included in the assay are determined, multiplex assay based on multiplex PCR of bisulfite converted DNA can be designed using several approaches. In the forensic field, methylation snapshot has been preferentially used for multiplex construction because of its multiplexing capability and the same workflow used in SNP analysis. In our previous slide, we could see the snapshot multiplex that allows the simultaneous analysis of 9 CpG markers. The multiplex assay, when the design is completed, is validated using independent set of samples, and interpretational guidelines will be established to report the assay result. Now I want to get into talking about the identification of body fluid specific CpGs. We produced epigenome-wide DNA methylation profiles of 42 body fluid samples, including semen, blood, saliva, vaginal fluid, and menstrual blood, using Illumina's human methylation 450 village array. To identify CpGs with body fluid specific differential DNA methylation, the mean value of the average beta score was calculated for every CpG in each type of body fluid, and they were compared among the body fluid. Firstly, the CpGs that exhibited more than 30% discrepancy in the mean values of average beta scores between a certain type of body fluid and other types of body fluids with a p-value less than 0.05 in a student's t-test was selected. In the case of semen, many CpGs showed 30% or more discrepancy in DNA methylation from other body fluids, and the comparison of vaginal fluid and menstrual blood did not show any significant CpGs. Therefore, we decided to select the semen-specific markers first by comparison of semen and all other types of body fluid with a cutoff value of 50% or more discrepancy. Then we excluded semen and took vaginal fluid and menstrual blood together to select markers that are specific to other types of body fluid. Then we further extracted markers that show body fluid specific hyper or hypomethylation, and at the same time complete non-methylation or methylation in other types of body fluid. In addition, we made sure that these markers have a low standard deviation in the same type of body fluid. If we look at the semen-specific marker candidates from the array data sheet, you can see that the mean value of semen is around 90%, and the mean value of DNA methylation in other body fluids is around 3%. And on the right side, you can also confirm that the standard deviation of semen and other types of body fluids are all less than 10%. This slide shows DNA methylation profiles of 64 body fluid specific CpG marker candidates. Each row indicates markers and each column indicates body fluid samples. Red color indicates hypomethylation and green color indicates hypomethylation in comparison with the average methylation of the samples. Here we can see that the same type of body fluids cluster together based on the DNA methylation at the selected markers. At this point, I'd like to highlight the importance of on-off signal in selecting body fluid-specific marker candidates. Here we can easily see that the body fluid identification is possible even in mixed samples. This is attributed to the fact that the assay consists of markers with an on-off signal. Showing on-off signal does not mean just having a high discrepancy in DNA methylation between target body fluid and others. It does mean only target body fluid shows positive signal for methylation or non-methylation. Therefore, these markers show methylation signal only in a target body fluid with almost complete non-methylation signal in other types of body fluid, or show non-methylation signal in a target body fluid with almost complete methylation in others. These characteristics facilitate the body fluid identification even when the body fluid-specific methylation or non-methylation might be diluted in mixtures. So selected body fluid-specific CPGs show high methylation signal only in a particular type of body fluid with a low standard deviation for the same type of body fluid. If you look at these bar plots, you will see that the semen-specific markers SC1 and SC2 demonstrated high methylation signal only in semen samples, while specific markers BL1 and BL3 showed high methylation only in blood. Vagina fluid-specific markers BF1 and BF2 showed high methylation signal only in vaginal fluid and menstrual blood, and saliva-specific marker SA1 demonstrated high methylation signal only in saliva samples. Because the previous analysis could not differentiate between vaginal fluid and menstrual blood, we tried again to select menstrual blood-specific CPG marker candidates by comparing 3 vaginal fluid with 3 of each menstrual blood obtained from the first, second, and third days of menstrual bleeding. This time, we could obtain several CPGs with 30% or more methylation difference between vaginal fluid and menstrual blood collected from a day of menstrual bleeding. A total of 15 CPGs were finally selected as a candidate, and their menstrual blood specificity was confirmed by comparison with array data obtained for other body fluid samples. Among 15 CPG marker candidates, 3 were located in the promoter region of the SLC26A10 gene, and 2 of them showed high menstrual blood specificity. After selecting body fluid specific marker candidates, the body fluid specificity of the selected markers were further investigated using targeted bisulfite sequencing. In the targeted bisulfite sequencing using methylation snapshot, a blue peak represents the nucleotide G as a methylation signal, and the green peak represents the nucleotide A as a non-methylation signal. Therefore, body fluid specificity of methylation markers could be confirmed by the presence of a blue peak in a target body fluid. Here, blood markers show high blue peaks only in blood samples, and semen markers show blue peaks only in semen samples. Before we get too far into it, I want to deal briefly with bisulfite conversion and DNA methylation detection using methylation snapshot. As you can see here, bisulfite treatment converts unmodified cytosine to uracil. Therefore, this procedure, followed by PCR amplification, can reveal the methylation status of every cytosine by comparison of sequence information between the reference genome and bisulfite-treated DNA. Once we get a PCR amplicon from bisulfite-converted DNA, from which we can measure DNA methylation using various methods. So I will show you how to design PCR primers that target bisulfite-converted DNA. For convenience sake, I will talk only about the most typical program as primer. To use this program, you can just paste the genomic DNA sequence and indicate the location of the target CpG site. At this step, we can select one of the two strands, that is original top strand or original bottom strand, for sequence analysis to measure DNA methylation. Because the converted DNA strands are no longer complement each other and present as a single strand, we generally select and amplify one of the two single strands. Besides, a strong point of this program is that it defines the minimum number of non-CpG in a primer, because all cytosines at non-CpG should be converted to uracil by bisulfite treatment with a default setting of a minimum 4 non-CpG. We can get primers that will selectively amplify bisulfite modified DNA with unmodified or incompletely modified DNA left over. As an output, we can get the CpG island information on the sequence as well as several PCR primer pairs specific for bisulfite converted DNA. At this stage, we can locate candidate PCR primers on the in silico bisulfite converted DNA sequence. Let's have a look at the primer binding site. In the forward primer, 7 small T's derived from non-CpG are present, and in the reverse primer, 4 small T's are present. So this primer pair will not amplify unmodified or incompletely modified DNA. Primer binding site does not include a CpG site, but PCR fragment includes several CpG sites that allow DNA methylation measurement. Then I will show you how to measure DNA methylation using methylation snapshot. Methylation snapshot generally refers to methylation-sensitive single nucleotide primer extension. The chemistry is based on the dideoxy single base extension of an unlabeled oligonucleotide primer. In this slide, PCR amplified bisulfite converted DNA includes CG or TG sequence in the forward strand and CG or CA sequence in the reverse complement strand, depending on the methylation status of the cytosine of the CpG site. If we design a SB primer in reverse direction, the primer binds to a forward strand and the polymerase extends the primer by one nucleotide, adding a single dd-GTP or a single dd-ATP to its 3' end. If we design an SB primer in forward direction, the primer binds to a reverse complement strand and is extended by one nucleotide, adding a single dd-CTP or adding a single dd-TTP. The fluorescent color readout reports which base is added, and we can calculate the methylation percentages from nucleotide intensities. As I mentioned earlier, the multiplexing capability of the snapshot reaction allows us to incorporate multiple CpG markers in a single reaction. After multiplex PCR which targets multiple CpG markers, we can carry out multiplex SB reaction using primers with different lengths. SB primer length can be adjusted by adding polynucleotide tails to the 5' end of each SB primer, and the length of tailed SB primers determine the order of fluorescence signal for a marker in the electrophotogram. To construct the snapshot multiplex you see here, we use SB primers with a range from 21 to 88 nucleotide in length, which was adjusted by adding poly T-tail to the 5' end of each SB primer. This snapshot multiplex enables simultaneous analysis of non-CpG markers, and methylation levels at multiple CpG sites can conveniently be observed at a glance in the electrophotogram. In order to introduce DNA methylation-based body fluid identification in your laboratory, you can download a detailed protocol for this snapshot multiplex from our website. Or you may carry out independent validation for reported markers and can construct a multiplex assay on your own. The authors of this paper, shown here, evaluated certain previously reported markers in German body fluid samples and selected SA1, BL1, BF2, and SA1 markers from our multiplex as robust body fluid specific CPC markers for multiplex assay construction. Here they suggested that a combination of only those 4 highly informative markers would be enough for clear body fluid identification. Now that we've covered identification of body fluid CPC markers and construction of a snapshot multiplex, I'd like to talk about how to set up DNA methylation profiling in your laboratory in detail. When you want to introduce a certain method in your lab, the first thing you do is referring to previous articles. Here I show you our 2 previous publications. The first paper is about a collaborative validation of a snapshot multiplex for simultaneous detection of 7 CPCs for the identification of blood, saliva, semen, and vagina fluid. And the second paper is a more recent one and is about a snapshot multiplex for the detection of the same 7 markers plus 2 CPCs specific for menstrual blood. Once you decide which multiplex you would set up, you can prepare the reagent and kits that are needed for the assay referring to the article. Because the assay we are interested in is characterized by the analysis of bisulfide converted DNA through single nucleotide primary extension process, it needs a bisulfide modification kit, bisulfide converted DNA specific primers, and the snapshot kit for SB reaction. Other enzymes and reagents are something that simply can be seen in the forensic laboratory. As I mentioned earlier, our assay actually has the same experimental procedure as the SNP scoring using the snapshot kit, except we used bisulfide converted DNA as a PCR template. So the right selection of bisulfide modification kit is important for success in the following experimental procedure. Several commercial kits are now available, and each of them has a different capacity in the optimal or minimal quantity of DNA. You can select appropriate modification kit based on recommended or minimum input quantity of DNA. Once you figure out what the snapshot multiplex for DNA methylation-based body fluid typing looks like, you can order primer and perform tests. However, the whole process is composed of several complex tasks, so it would be nice to start with a single source and sufficient amount of DNA. At the first attempt, I recommend to use 50 to 200 nanogram of genomic DNA for bisulfide conversion. After bisulfide conversion, we should note that bisulfide converted DNA is present as a single strand, so it will be stable only for 1 day at room temperature, 1 week at 4 degrees, and 2 to 4 months at -20 degrees. If the bisulfide converted DNA is prepared, we can proceed to multiplex PCR and SBE according to the protocol. Because it will be the first attempt too, I recommend to use enough amount of bisulfide converted DNA, such as 10 nanogram or more converted DNA. We generally do not quantify bisulfide converted DNA, so the approximate amount of DNA can be estimated from the portion of the evaluate of bisulfide conversion reaction. This experimental procedure of multiplex PCR and multiplex SB are not different from general SNP scoring method. If you have an experience in mitochondrial DNA SNP typing using the snapshot kit, the whole process would be pretty much the same. Then, how do we interpret electrophoregram after capillary electrophoresis? Once you get an electrophoregram after snapshot multiplex reaction, You can designate positive peaks if the signal intensity is over the detection threshold. In our case, 100 RFU was used. So, when you first establish setting for CE analysis on your DNA analyzer for snapshot reaction, you have to make sure all of the expected peaks are over this threshold. However, these values can be varied depending on your DNA analyzer. If you designate positive peaks, you can put peak height values into the Excel sheet and then calculate methylation percentages. Because all of the SV primers in our multiplex were designed in reverse direction, the DNA methylation percentages are obtained by dividing nucleotide G intensity corresponding to methylation signal by nucleotide G plus nucleotide A intensity corresponding to non-methylation signal. This electropherogram displays the data with more than 70% methylation at 2 vaginal fluid specific markers and 0% methylation at the rest of 7 markers. To determine a body fluid type, we should determine minimum threshold for positive methylation signal and then check which CPGs show the methylation signal in a sample. Generally, we can say that the methylation signal is present when the height of the blue peak is over the detection threshold. However, unexpected methylation signal with less than 10% has been observed from the same batch of bisulfite converted DNA in 2 of the 6 laboratories of the collaborative exercise. Therefore, the methylation percentage less than 10% was ignored. Then body fluid type can be determined by comparison with the reference DNA methylation profiles. If you think of our snapshot multiplex, each 2 markers are specific for semen, blood, and vaginal fluid, but only 1 marker is specific for semen and saliva. So, if 2 markers are positive for semen, blood, or vaginal fluid, we can say that the body fluid is observed. But if only 1 marker is positive for semen, blood, or vaginal fluid, then we would say it is not reliable. With regard to saliva, only 1 positive methylation signal as saliva-specific marker can indicate the presence of saliva. However, more careful considerations will be required when additional methylation signal appears at other CPG sites. The time we analyze saliva-specific marker assay 1 in more samples, we could observe 2 vaginal fluid and 1 menstrual blood samples with a methylation signal at the saliva marker in addition to vaginal fluid or menstrual blood specific marker site. After all, additional specific markers for saliva are needed to the multiplex for better body fluid typing analysis. Interpretation of menstrual blood profile is more complicated. Actually, menstrual blood samples show different methylation profiles depending on the menstrual cycle and the sampling method. As you can see here, all menstrual blood samples basically show methylation signals at 2 vaginal fluid specific markers, and many samples show the methylation profile similar to vaginal fluid. However, the 2 menstrual blood specific markers show methylation signal only in menstrual blood. So, if both of the menstrual blood specific markers or one of these 2 markers and 2 blood specific markers show additional methylation, we say that menstrual blood is observed. Moreover, when the samples were collected from the inside the vagina rather than from closed or sanitary path, methylation signals are strongly observed as vaginal fluid specific markers. Lastly, for clear understanding of our system, more research may be needed using more samples. A test with more body fluid sample is needed for independent validation and establishment of interpretational guidelines in your laboratory. Another test with serially diluted DNA is needed for having information on the lowest amount of DNA that can produce reliable results. and other tests with mixed samples would be needed to get a good perspective on the future joint interpretation with SGL and DNA methylation profiles. After setting up the experimental conditions and interpretational guidelines that are required for reproducible body fluid typing results, you may be able to develop reporting guidelines and establish SOPs. Now let's move on to the last part of my presentation, casework sample analysis. Casework sample analysis was performed in the National Forensic Service of Korea according to our protocol. The first case that I will show you is a rape case where the perpetrator was a friend of the victim's boyfriend and they stayed up late drinking together that night. A vagina swab collected from the victim was positive for semen in a CP test and showed autosomal STR profile of the perpetrator. A penile sample was also collected and showed STR profile of the victim. A stain from the belly of the victim was negative for semen and saliva but showed STR profile of the perpetrator. The NFS researcher performed DNA methylation-based body fluid typing using the remaining DNA from the penile swab and a stain on the belly. As you can see here, penile swab showed DNA methylation profile of vagina fluid and the stain from the belly showed DNA methylation profile of semen even though it showed negative for semen in a CP test. Particularly noteworthy is the fact that the amount of DNA used here is considerably low and the peaks are designated with a lower detection threshold such as 50 RPU. The second case is a distal penetration occurred on the bus. The perpetrator happened to sit next to a woman and sexually abused her with his hand. Vagina swab was negative for semen and showed only STR profiles of the victim. A swab wiped the fingers of the perpetrator showed STR profile of the victim, but there had been no way to know the body fluid source of the DNA until methylation-based body fluid typing was performed. Based on the presence of the blue peak at two vagina fluid specific markers, the DNA was considered to be originated from vagina of the victim. DNA from the perpetrator was not detected from the swab, but there was a need to examine DNA methylation profile of the skin. In our previous multiplex was tested on the skin swab. Among 8 markers, 6 indicated in red overlapped with the markers from our present multiplex and all showed non-methylation signal from skin DNA samples. The third case is about the murder occurred in 2004. A female karaoke worker was found to be dead, and a knife and an unknown stain was found near the victim. From the test in 2004, knife blade was negative for blood but showed STR profile of the victim, and the stain was positive for saliva and showed unknown male's STR profile. Interestingly, the unknown stain showed methylation profile of saliva, but the knife blade showed DNA methylation profile of vagina fluid. However, when we investigated the story, we could know that the victim's body was damaged by cutting the left breast part and vagina with a knife. Therefore, we could say DNA methylation profile was consistent with the scenario of the case. More noteworthy part was that the DNA methylation profiling was successfully done with 13-year-old DNA samples. The last case is an indecent exposure with no suspect. Police received a call reporting that a man was indecently exposing himself at a bus station. When the police arrived there, the perpetrator ran away, but a stain could be collected as an evidence from the scene. The stain was positive for semen and saliva. The stain was positive for semen and saliva in a preliminary test and showed a two men's mixed STR profile. To separate the two men's DNA, NFS researcher performed differential extraction. DNA from precipitate show the men's ester profile, and the DNA from the saliva show the another men's ester profile. DNA methylation profiling was also performed in the DNA from precipitate as well as the DNA from supernatant and initial mixture DNA. If you look at these, you can see that the initial mixture stain showed methylation signals at 2 semen-specific markers and a saliva-specific marker. After differential extraction, the precipitate showed DNA methylation profile of semen and supernatant showed DNA methylation profile of saliva. However, since there was no suspect and the ester profile from semen did not match any one of the national DNA database, this case could remain unknown. So, we performed DNA methylation-based age prediction using the remaining DNA from sperm portion and got a result implying that the perpetrator is about 60 years old. After a while, the perpetrator was caught by CCTV analysis and found to be in his late 50s. These results demonstrate the high prediction accuracy of DNA methylation-based assay and support the addition of DNA methylation profiling to forensic DNA analysis. Finally, I'd like to thank everyone who have worked hard to make all this possible. Thank you for your attention. I will look forward to the Q&A session.
All right, thank you so much, Dr. Lee. We are going to move into our Q&A now. I'm going to switch everyone's layout. Before we do begin our Q&A, I want to make mention of our presenters' contact information down there at the bottom of your screen. So before you leave today, please make sure to take down their emails just in case you do have any follow-up questions that you'd like to ask. So we had several questions come in for both presenters. We're going to do kind of a back and forth. So we will first start with Dr. Vidaki. Let me get to our first question. As I said, we did have so many that came in. I need to scroll up. So our first question in the webinar paper, is the MAD of 3.34 actually comparable to the SE of 3.88 years in Hanam?
Thank you very much for this question. I would like to say that there are different ways to present the measure of variability or the variance of your prediction, and different studies and different models report their prediction error in a different way. Two of these is either, as mentioned, the mean absolute deviation or the standard error. So the standard error, which is pretty much the standard deviation, is simply the square root of the variance. However, the mean absolute deviation is a different measure that takes the absolute values themselves rather than the squares. So you can say that there are two ways to say the same thing, but if you would like to compare between studies, it would be nice that the same measure of variability is used. I would say yes, they are comparable, but the actual comparison should be done with the same measure.
All right, next question will be for Dr. Lee. Dr. Lee, Christian Taylor has asked, can this DNA methylation body fluid identification test be used in mixture analysis?
Thank you for this question. As I showed in my presentation, our multiplex can be applied to the mixture analysis because the markers used in the multiplex show enough signals, so even if the methylation, body fluid specific methylation could be diluted, that could be used for the identification of the specific body fluid.
All right, thank you, Dr. Lee. Dr. Vidaki, this one's for you. What was the improvement then for blood-only samples when moving from 3 to 5 CpGs? I take it all the samples for this test must be a single source. Is that correct?
Glad to answer this question. So in this specific study by Koch and Wagner, where they tried 2 different models with either 5 or 3 CpG sites. The actual difference was 1 year in the prediction error. So with 5 CpG sites, I had a prediction of 9.3 years, and then going down to 3 CpG sites, then the error increased to 10.3 by 1 year. So you can see that actually the contribution of different CpG sites, like we also saw in our study, is quite different. And we could have CpG sites that are absolutely meaningful and very important in the prediction, whereas some contribute only to a smaller degree. Now, the problem with the DNA methylation, answering the second part of the question on single source, it is that it is a quantitative value, meaning that it's a ratio ranging between 0 to 1. So it's a number. It's not qualitative as you would get with the SNP analysis, where you have categorical values, where you have A or T or C or T in this case, but you get a number. So it's very difficult to separate this if you do have mixed samples. So I think these predictions have to be single source at the stage that we are now.
All right, thank you, Dr. Vidaki. Dr. Lee, this one's from you from Jeffrey Thompson. Is there research planned on touch or skin DNA, such as quote-unquote as a body fluid source? This could be especially useful in some circumstances.
Yes, I agree with this idea, and we try to search the skin-specific methylation markers, and we could find several candidate markers from the public database, but actually we failed to enough amount of DNA from the skin samples, so we could not confirm the data using local specific analysis. So if we could enough amount of DNA for biostrophic conversion, we could test the body fluid skin specificity of the candidate markers afterwards. I think it would be it's better trial if we could get a more amount of DNA from the skin.
All right, thank you, Dr. Lee. Dr. Vidaki, again, this one's for you. Based on the reproducibility numbers that you cited, it seems that the test needs to be run many times to converge to an accurate prediction, as each individual prediction had very high error rates. Is this the recommended approach?
Thank you for this question. It's actually, yes, very interesting observation. So the numbers that presented were actually the same sample run 6 times using only 1 nanogram of DNA into the library preparation, meaning the lowest limit that we could detect a meaningful result. Therefore, we expect that with low amounts, the variability will be higher, therefore the reproducibility will be lower. Generally, when we were testing the first prediction results using the MySeq method, we saw that, as you perhaps expect with every quantification method, if you run things in duplicate or in triplicate, you kind of have a way to account for some technical variability, and the predictions were improved. I think with a single analysis, we had a 10-year error, and then with a triplicate analysis of the same samples, we had the error down to 7.45, like I presented. So yes, the recommended approach would be to run things in duplicate or in triplicate if possible. However, further testing needs to be done, not only to see how useful this is, also not only when you have very little amount of DNA, but also to assess the reproducibility of the final method, if possible, of course, and the sample quantities allow.
All right, thank you very much, Dr. Vidaki. So Dr. Lee, on the last question regarding touch or skin DNA as a body fluid source, Christian Taylor wanted to kind of elaborate on that and ask what the amplification amount would be required for it if it was a body fluid source.
Yeah, thank you for the question. Actually, DNA amount is really important for the DNA methylation analysis, and according to the recent paper by Jana Neuer, he says that the amount of the accurate DNA methylation profiling can be determined by the point what we would like to see. So actually, DNA methylation is a binary event. So and also the DNA methylation percentage represented the average frequency of DNA methylation in the all DNA molecules in a test tube. So the amount is really important, and it affects statistical respect. So the paper says that DNA methylation for body fluid, such as for semen identification, can be accurately done by smaller amount of DNA rather in comparison with the DNA methylation analysis for age prediction. And also our DNA methylation-based body fluid typing see the presence or absence of DNA methylation signal rather than exact quantity of DNA methylation. It needs much less DNA compared to the age prediction. But in any case, we usually use 10 nanogram of bisulfite converted DNA for reliable results, and sometimes, less than 2 nanogram of bisulfite converted DNA is successfully done in body fluid identification.
All right, thank you, Dr. Lee. Dr. Vidaki, this one's back to you. Have you tested any sample types besides blood and saliva? And do you think separate models will be needed for different sample types?
Thank you. This is a very interesting question. Well, so far we have been focusing our analysis in the probably two most commonly collected evidence from crime scenes, meaning blood and saliva. And as we saw using the CPG size that we proposed, perhaps it's possible to have a single model for both. However, things can get more complicated when we would like to expand this to other body fluids and other tissues. So generally, age-associated DNA methylation is expected to be found in all the tissues of our body. And as Horvath showed in his study, there is a subset, a small subset of age-associated CPG sites that show aging effects in all tissues. However, the aging effects and the rate of these effects and the size effects can be different. So I think that we might still would like to use the same set of CPG sites, but it would be very advantageous to have a separate model per tissue. It's interesting, I think, to expand in tissues like semen, as also Professor Huang Yang has shown in her recent paper, despite Horvath mentioning in his study that he did not find any age-associated effects in this tissue, and also in skin or other forensically relevant body fluids and tissues. So I think it is a matter of future investigations to see how robust and how tissue-specific these effects are, and what would be the best way forward.
All right, thank you, Dr. Vidaki. Dr. Lee, this one's back to you. Is there any research being conducted on tears, although it may only be found in trace quantities?
I couldn't see any research on tears. But it would be good if we can find the markers specific for tears afterwards. Thank you.
All right, Dr. Vidaki, this one is a three-parter, so you can tackle it as you would like. First part is, do you plan on identifying slow and fast-aging people, perhaps using a telomere test and testing their blood? Also, how common are slow and fast agers in the population, and is it possible they are the variability in the age predictions?
I would like to start answering this question by saying that what we detect with those epigenetic clocks is biological age rather than chronological age. We are looking the association between DNA methylation with how fast or slow our cells age. Now, of course, as we expect, and we've all heard this saying, oh, he has good genes, so he doesn't age fast, or the opposite. We know that there are some genetic effects on how fast or not fast we age. However, the purpose of an age prediction tool for forensic applications would not be to detect if somebody aged fast or not. The purpose, though, and what we would need still to address when we apply this into forensic casework is, can we assess statistically how likely it is that the person that we are looking for changes or affects our prediction because they age fast. Now, in their study in Hanem, where they did try to see and look into those few samples that were aging either very fast or very small, it will be difficult for me right now to put a number there, how often this is seen in populations. In Hanem's model, there were only a few samples out of the 660. However, they saw that when they tested various parameters and clinical factors, the gender was the only one that showed a significant contribution to the aging rate. As I mentioned before, they saw that the methylome of men appeared to age approximately 4% faster than the one of women. However, things like BMI, body mass index, did not show such effects. So I think it should be part of the validation of every method to test for those possible effects, including aging rates. And I think that was my answer.
All right. So the next questions we have came in during Dr. Vidaki's talk, but Dr. Lee, if you would like to jump in at any time, please feel free. So the question from Arthur Young is asking, wouldn't there need to be some consideration when testing cigarette butts of the effect of smoking on said markers? I know you'd mentioned cigarettes a little bit in your talk. For example, smoking may prematurely age smokers, and then Jared Rice piggybacked on to that, asking, would mutagens like smoke and narcotics laced with adulterants damage the viability of these markers for identification?
Thank you for this interesting question. Indeed, what we have presented so far and what people have published so far in this topic is using the ideal samples of pure single source body fluids, either blood or saliva or so on. So we know from some preliminary studies, especially using methylation to see for tissue effects, that indeed when we get to more complicated forensic type samples like minute blood stains or cigarette butts, the effects of methylation can be quite immense and could also affect the prediction, the age prediction, or whatever epigenetic-based prediction we are trying to do. I can give a small overview of my experience so far when working with the twin application. When I tried to see if we could get the same methylation levels in small blood stains compared to whole blood reference samples and also cigarette butts compared to saliva samples, the last not being published yet, but we hope that they will be in the first quarter of 2018. We saw that we had both technical variation there, but we also had the effect of other factors, including the cell composition and perhaps, as it was suggested here, smoking could have also an impact. So cigarette butts are being considered and are being very related to saliva samples, but one should also think that they do contain skin cells, because of course we smoke with the skin, we can transfer skin cells from our lips, which, as we saw from the talk of Professor Huang Yang, we could have differential DNA methylation there. So we need to find a way to assess those, and also if the age prediction is being planned to be built in saliva, also test the effect of smoking. Usually in medical research studies, age and smoking are considered as the top two covariates to account and normalize before even do any other analysis. So I would not be surprised to see that some, maybe not all, but some of the CPG sites, either being tissue-specific or age-specific, are indeed also influenced by smoking.
All right. Next question we have is from Angela. She's asking, has mitochondrial DNA methylation been considered as a forensic biomarker as well?
This is very interesting, and of course mitochondrial DNA methylation could be very valuable, as many people can think, because it's being found, mitochondrial DNA is being found in higher amounts and has been successfully applied to most challenging samples. This is a very interesting topic in the sense that the current literature is very contradictory. So there have been indeed a few studies, both medical and forensic studies, assessing the possibility of applying DNA methylation of mitochondrial DNA for the age prediction. Generally, though, it seems that the methylation, the CpG methylation of mitochondrial DNA is very, very low on a kind of a large scale. There are, I think, only something like 160, 170 CpG sites on the mitochondrial DNA, that most of them show methylation values below 10%. However, there was a study, a recent study that used only 2 CpG sites out of the entire mitochondria, and they could build a model that could explain some of the age variance with a prediction with a mean absolute deviation of 9.3 years. This is very positive, and perhaps future investigations will really determine how useful mitochondrial methylation as a marker can be. However, there have always also been studies showing technical variation. For example, there was a study that showed that because of its circular nature, bisulfite conversion might not be as successful and efficient compared to the linear nuclear DNA. So I think if somebody wants to test this, they would need to design a strategy to account for a lot of technical variation on top of the biological variation.
All right, and this is our last question for today. A great talk from both of the presenters. Fabio is asking, could you expand more on the potential application of age-associated DNA methylation profiling on mixture analysis?
Thank you, Fabio, for this question. I think that eventually we would like methods that can be used in mixed stains, because we do have a lot of this in forensic casework. As I mentioned briefly before in answering another question, mixture analysis is one of the kind of challenging points when you want to apply epigenetics and specifically DNA methylation due to its quantitative nature. However, there have been a couple of studies published on addressing the cell composition. There have been some studies mainly in buccal cells and saliva samples. People tend to assume that saliva samples or saliva stains contain only buccal cells, but we have seen also in our lab, but also from other studies, that the percentage of leukocytes that are present in our saliva by default because of gum bleeding or because of cell defense mechanisms can range from 10 to 50% of the actual saliva samples. So there, authors have tried to detect a very small number of CpG sites
that are very robust among individuals and can be used to assess whether you could separate blood cells for BACA cells and correct for that in your methylation analysis. So I would assume that if somebody wants to apply the age prediction in mixed stains, they could use a similar approach and use some set of CpG sites that are kind of known methylation in a way to normalize what you see. But I can tell that this will definitely be challenging, and it is a lot of testing before we are able to answer that question.
All right, thank you so much, Dr. Vadaki and Dr. Lee. We are going to go ahead and switch over to our closing now. You will notice in the bottom left-hand corner of your screen, we do have a PDF version of both of today's presentations. So make sure you download those directly from the room before you leave today. All you have to do is click on the title of the file and then click the Download Files button at the bottom of that file share pod. Before we do close out, I would like to hand things back over to Daniele Podini, our moderator, just to give us some closing remarks.
Thank you, Josh. Thank you, Athena and Han Yang. These were outstanding talks, and I learned a lot, and I feel there's a great response by all the attendees that I also thank. I just wanted to say that our next appointment with the SNP webinar series is on January 16th. We will be talking about ancestry prediction, but not only the analytical aspect of the prediction, but also how to deliver that information to investigators. And so the bias and the perception in the delivery of the information. It's going to be another great webinar. So thank you again to all. Thank you to RTI, and happy holidays to all of you. And back to you, Josh.
All right, thank you, Dr. Podini. And again, obviously we would like to thank our subject matter experts, both of our presenters today, Dr. Vadaki and Dr. Lee, for taking time out of their day to deliver this information to you, our audience. Obviously, we would like to thank all of our attendees for joining us today. These webinars are definitely not possible without your attendance and your support. Before you leave today's webinar, we always ask that you participate in our short survey. You'll find it there in the center of your screen. Simply click the word "Here" and it will pop up a new window for you. And again, just go through our quick survey. We want to make sure that we deliver the content and disseminate this information out to you, our audience, in the best way possible. And this survey provides us very valuable feedback. So we would greatly appreciate it if you would take a few moments before you leave. Also popping up on your screen is a new browser window for our website. Please go ahead and check out our website. All of our new course listings, all of our archival webinars that we have hosted in the past are there. Everything is at no cost to you. So please feel free to browse through our website and take any number of webinars that you would like to attend. Again, thank you so much, Dr. Vadaki and Dr. Lee, for your presentation today. Again, make sure that you do download the PDF versions of today's slides there in the bottom left-hand corner in the file share pod. I'll leave this room open long enough so that everybody has that opportunity. And then also in the chat pod over on the left, Janine has put some links to our social media pages such as Facebook, Twitter, and LinkedIn. If you'd like to connect with us on there, we always keep everyone up to date on all of our future course offerings through our social media. Again, thank you everyone for attending today. We hope to see you in our next SNP webinar, and we hope everyone has a great rest of your afternoon.